The purpose of this series of mini lectures is to give you an overview of the general histological structure of glandular epithelium, or more specifically exocrine glands. Glands are classified into two groups depending on how their secretion products are released. Exocrine glands secrete their products directly onto the surface or via a duct that is connected to the surface. The duct may or may not modify the content of the secretions. These glands will be the subject of this lecture series. Endocrine glands secrete their products, called hormones or chemical messengers, into adjacent interstitial tissue, to then enter the blood vascular system and interact with target cells at various locations in the body. These glands are covered in my lecture series on the endocrine system in the Histology of the Human Organs course. In this lecture series, it's not the intent for you to understand the full complexity of different exocrine glands, because, again, I describe details of many of these glands later in that Histology of Human Organs course. At the end of this lecture series, you should be able to recognize the presence of unicellular glands in an epithelium, particularly a goblet cell, and know that if the duct portion of the gland is unbranched, then it's a simple gland. If the duct portion is branched, then it's a compound gland. Know that if the secretory portion is shaped like a tube, then it's a tubular gland. If it's shaped like a flask or grape, then it's an alveolar or an acinar gland. And if it ends in a dilation or sac, then it's a tubulo-alveolar gland. Appreciate that tubular secretory portions may be straight, branched, or coiled. Alveolar portions may be single or branched. And so, knowing this terminology, appreciate the general classification and histological appearance of the multicellular glands listed below. And then be able to distinguish mucous and serous secretory cells and duct portions of glands.